Okay, good morning. Since I don't see the session chair, so I will just start by myself. Thank you for coming to my talk. And looks like there are many more people than last year. So this work done mainly by my student Ziyong Wang, but because of visa issue, he couldn't come here. So I will just present the work on behalf of him. So this slides just briefly introduce the problem I'm going to address. So in this talk, I'm going to talk about how to predict interresidual contacts. So here we use a simple definition. We just measure the distance between the CF or C beta atoms between two residues. As long as the distance is less than 8 angstroms, then we say there's one contact between these two residues. So this picture here show you a very small protein, and the contacts, the blue box represent the contacts. And so there are three contact categories: short range and medium range and long range, depending on the sequential distance of two residues around the primary sequence. The reason why we wanted to do contact prediction is because contact has been used like for protein alignment and protein classification. Currently also model describe the relationship between two residues, like the spatial relationship or functional relationship. And contacts are also very helpful for protein structure prediction. Quite a few methods have been developed for contact prediction. For example, the number of methods have been developed based on the residual coevolution, like the first method, like the mutual information-based method, and the two recent methods, cycle and EVfold. These methods usually require a large number of homologous sequences. And if we don't have many sequence homologs for the protein under consideration, then this method usually don't have very good performance. And the cycle and EVfold, they are better than the mutual information because the former two methods differentiate the direct and the indirect residual couplings. And the cycle and EVfold also enforces sparsity, since we know the contact map matrix actually is very sparse. And the second category is the machine learning method, the supervised machine learning method like NNCon, neural network-based method, and SVM-con, and recent CMAP Pro. So this machine learning method integrate several kinds of information like the mutual information and the sequence profile and also other information. But they usually predict the contacts one by one and ignoring the correlation between contacts, and they don't consider the direct and indirect residual couplings. And the third category is the first principle method. And this method doesn't use the rich information, and they do prediction by minimize the contact potential and also enforce some physical feasibility, like to try to produce a contact map which is physically feasible. And so in our work, I'm going to focus on proteins with a few sequence homologs. Since usually for proteins with a large number of homologs, we can find very good templates in the PDB. So which means usually we don't need to do a contact prediction when a protein have a large number of sequence homologs. So we focus on proteins with a small number of sequence homologs. And our method will try to integrate many different information, like we will integrate sequence profile and residual coevolution, and then also non-evolution information. We will also implicitly, we don't explicitly, implicitly differentiate direct and indirect residual couplings through better feature engineering. And finally, we will also enforce physical constraints which imply sparsity. And the right figure shows you the overall flow chart of our method. So first, we will use a machine learning method called random forest to predict the probability of any two residues forming contact. Then we will also combine this kind of prediction with some prior knowledge, like physical feasibility knowledge about the contact, and then combine these two kind of information and do an optimization, do a linear program optimization, and then generate a physically feasible contact map. So this is the overall of our method. This slide shows you the information used by our machine learning model. So first, we use evolution information in a single protein family, like we use the sequence profile, and we also use the residue co-evolution information. We use 2 types of mutual information here, which will be detailed later. And then we also use the non-evolution information from the derived from all the solved structures, like we use the residue contact potential. And we also use the mixed information derived from both evolution and non-evolution information, like we use the homologous pairwise contact score, and we also use the new statistical potential, EPAD. Finally, we also make use of the amino acid physical chemical properties. So all this information are integrated by our machine learning model, random forest. Here, briefly tell you a little bit about the mutual information we used. So we don't use the traditional mutual information. The first mutual information we use is the contrastive mutual information. So this kind of information will remove the local background by measuring the mutual information difference, one pair with its neighbors. And the second type of mutual information is actually, this is related to the inverse of the mutual information matrix. But we don't calculate the inverse directly. We just calculate the mutual information matrix and the square of the mutual information and the cube of the mutual information matrix. So we just use these 4 different mutual information to approximate the inverse of the mutual information matrix. So here I show you an example of the contrast mutual information. And in this picture, the up triangle is the mutual information matrix, and the lower triangle is the contrastive mutual information, and the blue box are native contacts. So you can see the mutual information itself contains lots of noise. And by using the contrastive mutual information, we can reduce some noise. And this slide tells you how to calculate the homologous pairwise contact score. So this is quite simple. Basically, we just calculate the probability of 2 residues forming contact between 2 second structures. Like here, we consider, so given for the protein under consideration, first we run sciblasts to generate a multiple sequence alignment. Then we consider all the homologous in the multiple sequence alignment. So we enumerate all the homologous and then calculate the homologous pairwise contact score. Okay, so finally, we train our random forest use data set, which is chosen before CASP10 started. And so in total, we use about 900 non-redundant protein structures. Any two of them share less than 25% sequence identity. And we use all the contacts in these training proteins and about 20% for non-contact pairs. And in total, our model has about 300 features. And we use 500 trees to build the random forest, and we do 5-fold cross-validation to make sure there's no overfitting. Okay, so after I just told you how to develop the random forest model to predict the contact probability of between any 2 residues. So after we have such a model, then intuitive is we just want to pick up those contacts, pick up those residue pairs with a high probability as our prediction. But actually, if you do this, then the contact map generated by the method may be physically infeasible. So we also want to make sure the contacts chosen by us are physically feasible. So this is the second component of our method. So here we use Xij to indicate just one contact between 2 residues I and J, and use R, the capital R, to represent a relaxation variable for some soft physical constraints. And the function GR is a penalty for violation of the physical soft constraints. In addition, we also have some hard physical constraints which shouldn't be violated. Okay, so then we just want to maximize the accumulative contact probability while minimize the violation of physical constraints. So we have such a linear objective functions here. So we want to maximize this linear objective function. And we use this kind of constraints. So the first soft constraint is the number of contacts between two second stretch segments is limited. So we just do some simple statistics and we find out, for example, for the helix to helix, 95% of the helix to helix pairs, they have less than 5 contacts. And the maximal contacts between two helix segments is 12. So for this kind of, so we just use a soft constraint to describe this kind of physical constraints. And we can also up and lower bound the number of contacts between two beta strands. Like the left figure show you the lower bound of the number of contacts between two beta strands. So here, the figure shows you some statistics, and the second structure is the predicted second structure, is not the real second structure. And the right figure shows you the upper bound for the number of contacts between two beta strands, and you can see the upper bound actually is linear, almost linear with respect to the length of the beta strand segment. And the third soft constraint is about the loop. And so simple statistics also show that only about 3% of loop segments have a contact between the start and the ending residues. So all these constraints are formulated as linear constraints. Then we add these linear constraints into our linear program. And we also have some hard constraints. For example, for the parallel contacts between two beta strands, the contacts of labeling residue pairs should satisfy these constraints, which means if the two labeling pairs have contacts, then this residue pair, the residue pair in the middle should also have a contacts. And similarly, for the anti-parallel contacts, we also have a similar constraints. And we also know for one residue cannot form too many contacts. Like one residue cannot form contacts with both J and J plus 2 when J and J plus 2, these two residues are in the same upper helix. So we can formulate these constraints as a hard constraint. And then we also know one beta strand can form beta strands with up to 2 other beta strands. So we have this. We can also represent this hard constraints as a linear constraint. So finally, we use this linear programming to, we just saw this linear program to do the contact prediction. Now I'm going to show you some experimental results. Okay. So we use, I just told you the training data we used. So we use 2 test data sets. One is the CASP10 test data sets. It has 123 proteins. As I told you before, so our training data set was chosen before CASP10 started. So by using CASP10 data set, we just want to make sure there's no overlap between the training and the test data sets. So among these CASP10 data sets, 36 are hard, which means these tau proteins don't have very similar templates in the protein data bank. And we also make sure all these test proteins, they have no sequence identity, so which means there's no redundancy among these test proteins. And so we also run BLAST, compare the CASP10 with our training data. And so the sequence identity between these CASP10 targets and our training data is very low. And the second set is a large set, so we call set 600. In total, it has 600, about 600 proteins. So this set also has no redundancy with our training set, and also has high resolution and at least 50 amino acids in each protein. And they should also have a little bit, at least a small number of predicted second structures, at least 5 amino acids with a predicted second structure being alpha helix or beta strand. Okay, so here is the first result I want to show you. So here, this slide shows you the prediction accuracy with respect to the number of sequence homologous. So given a protein under prediction, we can run like a CyBlast to detect all the homologous, and we can calculate the number of non-redundant sequence homologous proteins. So here we use MEFF to represent the number of non-redundant sequence homologous for this protein. Then we can divide all the Cas10 targets into some groups by MEFF, and we just consider the accuracy of the predicted contacts in each group. So this figure shows you the performance of a number of methods, including our own method called PHY CMAP. This is our own method. And also have the results from the two recent methods, CyCoV and EVFold. So these two methods are based on the residue co-evolution. And another two machine learning method, like NNCon, which is a neural network-based method, and CMAP. CMAP is a very recent machine learning method. So from this figure, you can see when MEFF, when we have lots of sequence homologous, like over 100 non-redundant sequence homologous, we can achieve pretty good accuracy. And so the CyCoV also performs very well when we have a lot of sequence homologous. However, when the number of sequence homologous is smaller than 100, then the residue co-evolution-based method doesn't perform very well, usually perform worse than the machine learning method. And our method performs very well across the whole range of the MEFF, especially when the MEFF is between 10 and 100, and our method also has a reasonable prediction accuracy. The prediction accuracy is above 0.5. So when the number of sequence homologous is between 10 and 100. And so in the following slides, I'm going to show you some more detailed comparison. So here I show you the results on Cas10 for medium range contacts. So we can compare our method with CMAP, a machine learning method, and the CyCoV is a residue co-evolution-based method. So from here, you can see our method performs much better than the CMAP and the CyCoV on the Cas10 targets. So this is results on all the Cas10 targets for medium range. And here is the result for the long-range contacts. Long-range contact prediction usually is much harder than the medium-range contact prediction, but still, our method overall, our method is better than CMAP and CyCoV here. Although the advantage is not as large as the medium-range contact prediction. So these are the performance on all the targets. Now, let's look at the performance on the hard targets. Since for hard targets, usually we don't have many sequence homologous. So from here, you can see when we don't have many sequence homologous, the residue co-evolution-based method usually the performance is not great, it's very bad. But the machine learning method like CMAP performs very well, and our method, because we use much more information than CyCoV. Here is another slide to show you the performance on another big set. Here we only consider those proteins with less than 100 non-redundant sequence homologous. And the left figure compare our method with CMAP, and the right figure compare our method with CyCoV. And in this picture, you can see when the number of non-redundant sequence homologous is less than 100, CyCoV usually just doesn't apply. So when we don't have many sequence homologous, it's better not to use the pure residue co-evolution-based method. Okay, so this is the average accuracy of our method. Now I'm going to show you some specific examples. And here, show you one cusp target. And so for this target, we don't have many sequence homologous. So MEF is equal to 31. And Right up triangle show you the native C-beta context, and left low triangle show you the prediction by our method, and the right low triangle show you the prediction by UV-fold. UV-fold is another residue co-evolution-based method. So for this target, you can see UV-fold just doesn't apply. So although our method is not very good, but is much better than the UV-fold for this target. And here is another cusp target. So for this target, we have lots of sequence homologs. So MEF is over 2,000. But still, for this target, our method perform much better than UV-fold. For example, so our method can achieve L over 5 accuracy is 0.74, but the UV-fold accuracy is only 0.419, so it's much lower than our method. And if you look at the picture, you can see it's hard to tell. If you look at the right picture, you can see it's hard to tell. You can see the prediction by UV-fold contains lots of noise. It's hard to tell which one is the contact. And here is another example. So for this protein, we even have this large protein, and we have more than 3,000 non-redundant sequence homologs. But even though there are so many sequence homologs, the right figure shows you the UV-fold prediction accuracy is not very good. But our method can generate pretty reasonable prediction accuracy. And finally, show you one more example here. For this protein, we have 1,800 sequence homologs, and our method has accuracy 0.9, and UV-fold has accuracy about 0.5, and it's hard to tell. So look at the picture, you can see the prediction by UV-fold is not very good. Yeah. Okay, so I just show you some example, and so finally I will just summarize my talk. So the first take-home message is we just couldn't simply use a single information source for contact prediction. So it's better for us to combine different information sources like a sequence profile and residue co-evolution, and also some non-evolution information. And so by doing this, we can achieve good accuracy even for proteins with 10 to 100 sequence homologs. And the second take-home message is the physical constraints are helpful for protein with a few sequence homologs. So the right figure shows you the difference, performance difference of our method when we use physical constraints or when we don't use physical constraints. So you can see by using physical constraints, it helps for proteins with a few sequence homologs. Okay, so finally, I want to thank my student. Actually, the major work is done by him. So the major credit should go to my student, and also thank the funding agencies. And you can also try our method through our web server. Okay, thank you.
Okay, we just have time for one question.
You sort of, well, it's very clear, thank you. The only question I had was if you could describe more the training of the random forest. What features are actually being input to the random forest?
Yeah, actually, we have some results in our paper about which features are important. Yeah.